words today because I want us to jump right into the eye right after we were. So make sure you fill out that attendance for me so I know that you are here. But where we finished up last class, we had just looked at some of the external structures of the eye. And remember, the external structures of the eye were all about doing what for the eye? You want to remember? Communication, right, were the two big ones. We looked at the eyebrow, we looked at the eyelids. We called the eyelids, what did we call them? The fancy word was palpebrae. Very good, upper and lower. And what's the muscle that controls the upper palpebra? Levator. Palpebrae superioris. Very, very good. We said the eyelashes were unique because every single eyelash is attached to a nerve. That's how we know when something approaches our eye or comes near our eye. And so what was the structure in the eye that produces this milky, oily fluid that helps lubricate the eye? Lacrimal caruncle. Very, very good. Wonderful. All right. So a few more external features of the eye that I wanted to look at before we go inside the eye. First and foremost, the eye, remember, is located inside the orbit of the skull. So there's a deep socket called the orbit that houses the eye. Only about 1/6 of your eye volume is visible from the outside. So when you look at your neighbor and look at their eye, you're only seeing about 1/6 of their total eye mass. The rest of that orbit. Why do you think there's only a portion of the eye visible on the outside and the rest is deep inside? Protection, absolutely. This is really vulnerable in our face and our body, and so we want to keep it safe and locked inside that bony mass. And if you look at this eye, what do you notice is encapsulating the eye? What is it sitting inside of? A lot of what kind of tissue? Yeah, a lot of adipose tissue, a lot of fat tissue. This orbital fat, the subcutaneous orbital fat, is there on purpose. Why do you think we have all this fat there? For protection and cushioning. Yeah, cushioning is the big thing here. So all that fat is very important for protection and cushioning. You can really tell if someone is malnourished because their eyes look sunken, right? That means their body has to metabolize that orbital fat, because your body wants to protect the eye. The last structure I wanted to point out to you here is this very, very thin white membrane that covers the inside of the upper palpebra, the whole front of the eye, and the inside of the lower palpebra. And that is called the conjunctiva. The conjunctiva. That word is right here, conjunctiva. This word should make you think of one thing. Conjunctivitis, which is pink eye. I'm sure you've had it. If you haven't, then lucky you. I'm sure you've seen it at least. The conjunctiva is a very thin, single layer of simple squamous epithelium that lines the eye, the anterior portion of the eye, and the inside of both eyelids, all to protect and lubricate the eye. Now, the eye is a very vulnerable spot physically because there's no bone on the outside, right? But also structurally in that there's not immune cells in the eye. So if you do get some kind of infection or bacteria or some kind of abrasion on the eye, all of a sudden it's very important and scary because there's no way for your own body to protect you and fight off that infection. So it's one of those few places in the body where there's no immune cells. So when that covering of the eye gets infected, we call it is a real thing that is super contagious across the way. Now, what else do you kind of notice already in this picture is attached to the eye? Lots of what? Lots of muscle. There are lots of muscles attached to the eye. I'm going to show you each one of those. Now, the main lubricating fact or piece of this picture all comes from one gland called the lacrimal gland. The lacrimal gland is up here kind of on the surface of your brow, I guess, is that lacrimal gland. Lacrimal glands all day, every day produce tears. It's a salt water fluid that's going to constantly bathe the eye. And it's going to produce upper, lateral edge of the eye. They wash across the eye from lateral to medial. The lacrimal caruncle kind of collects those tears. And those tears eventually drain down into the nose and back down the throat. But those tears are going to wash and clean and cleanse the eye all day, every day. And inside tears, we have one of our very first pieces of the immune system, lysozyme. Lysozymes are microbial enzymes that are going to help us clean our eyes all day, every day. So though we don't have any immune cells protecting us, bless you, in the eye we do have lysozyme that's produced by the lacrimal gland and constantly bathes across the surface of the eye and drains down. Now normally you don't notice tear production. This happens all day, every day, and keeps your eyes moist and ready to go. But every now and then we have an overproduction of lacrimal gland secretion. They spill over the lower palpebra, and that is crying, right, or your eyes watering. Either you got something irritating in your eye and that caused and triggered the reflex to produce more tears, or you're watching the Hallmark Channel, or like me, like a commercial right now, will send me over the edge and cause overproduction of tears from that lacrimal gland, and then, you know, you're a watery mess. But yeah, regardless, they're producing all day, everyday tears, and usually absorbed at that same space. Now, there are 6 different skeletal, yes.
So, if it's typically in our eyes, then why do we cry when it's bad?
So, part of there is a limbic emotional connection that triggers the lacrimal gland secretion that increases that tears whenever you cry. There's no physical cry when we're sad, other than the emotional connection with our brain. But yeah, there's no protective reason. When you cry when you're hurt, especially if you've had something like irritating in the eye, makes more sense, right? Otherwise, that's just a complex fun factor of us having emotions and annoying emotions, right? That we can't control. But that's a really good question. Yes, ma'am.
Why do you cry when you throw up?
I don't cry when I throw up. Do you? Oh, my eyes do? Well, okay. I thought you meant like.
No, like.
Okay. So think about the oral cavity, right? Let's go back to this picture. So, wait, what, did I miss something? No, okay. So when things are reversing this direction, right? There's lots of fluid coming. This is getting a fun place on a Wednesday morning. But so think about we're connected to the oral cavity here, right? The nasal cavity is connected superiorly to that oral cavity. So when things start happening, to also help flush, right? So when the body presses the massive eject button, lots of things are ejected. Get it all out, right? Get rid of everything. Christina, was that your question, too?
No. I was just going to ask you to repeat the function of lysozyme.
Oh, antibacterial, antimicrobial. Yes. It's our first glimpse into our innate immune system, the stuff that we don't even have to work at that just protects our body. Mm-hmm. Thank you for stopping me. And thank you for both of your good questions. I'm going to have to revisit crying and bearing up. Okay, there are 6 skeletal muscles that control the eye. 6. And we call these extrinsic eye muscles because these are not voluntary. They're not smooth. These are skeletal muscles that you can control. Now, I say they're skeletal that you can control, but often you don't think about where your eyes are moving, right? Often our eyes move reflexively and follow triggers like light or sound or movement. So they're reflex control, but they are skeletal muscles. So holler one of the muscles you see up here. We'll count all 6. Superior oblique. Superior rectus. Lateral rectus. Inferior rectus. Medial rectus. And then there's an inferior oblique as well. So there's 6. There's superior and inferior oblique, and lateral, medial, superior, and inferior rectus muscles. And you can see the eye is literally like a golf ball that can be tugged in every single direction with these skeletals. Let's look at the different directions this eye can be moved. And also here in this picture, you can really appreciate how the big mass of the eye is behind the orbit, right? And this big cranial nerve right here, cranial nerve number, not 1, 2. Remember, we don't smell through our eyes. We see through our eyes, and optic too. All right, good. Let's look at each of these muscles. So you've got superior rectus up here, inferior rectus down here, lateral and medial rectus. The superior oblique is this one right here that you can't really see. It's kind of tucked here, and the inferior oblique is here. And I've got written for you down on this slide how each of these muscles act on the eye. So the lateral rectus pulls the eye to the lateral side of the face. So when you look left, if you take your finger and put it on your nose, that's your medial rectus, making you cross-eyed, pulling that eye in. The superior rectus looks up and medial. That's your superior rectus. Your inferior rectus would be looking at the tip of your nose, or like down at your chin if you could see your chin. That's your inferior rectus. And then your obliques move your eye up and out and down and out. If you've got the PowerPoint printed out, those are reversed. This is true. The inferior oblique goes up and out, see it tugs on the eye here and points the eye up and out. The superior oblique tugs on the eye and points the eye down and out. So either way, you're going out but down by cranial nerves. Lateral rectus, anyone remember from lab, is controlled by, which is an A. Abducens, very good. The abducens cranial nerve controls lateral rectus. Think side eye, abduct the eye. Superior oblique is trochlear. Everything else is what cranial nerve? But not two, that's just sensory vision. Oculomotor. Oculomotor. So lateral rectus is abduction, superior oblique, oculomotor nerve, cranial nerve number 3. Cranial nerve number 3. So make sure you know those nerves for me and those different eye muscles. But we have, which is wonderful. Remember, this is our dominant sense, and our ability to move the eye helps us be alerted to when things are coming at us or moving around us, or lights are flashing around us and things are changing. All right, I hope that you printed out the picture of the eye on Blackboard, because we are going to draw. We are going to draw. So pull out that picture, wherever you've got it. We've got some really unique structures to meet here in the eye. Okay, that is a yes. Okay, so this is a cross-section of the eye. We're looking at it. It's sunk inside the orbit. You can see some eyelashes here at the front and the upper palpebra and the lower palpebra here. Tell me, what are some structures that you know? Because I know you know some of these structures already. Lens. Okay, so this right here is the lens of the eye. Right? And the purpose of the lens, I'm going to try to tell you the function and purpose as we go, is to focus light on the back of the eye. And the lens is going to be able to change shape as we focus things that are near or far onto the back of the eye. The lens is always growing and enlarges as we age. So it's got layers and layers of proteins, and the older you get, the thicker and bigger your lens is. So someone very young has a very movable and pliable lens, and a small lens. Someone very old is going to have a thick and I guess immovable lens. But these are just layers and layers of proteins, and these proteins are called crystallins. What's wild about the lens is it is transparent. We can see through it. You're looking through it right now as you're squinting to look at this tiny writing on this tiny screen, right? And these crystalline proteins are carefully arranged so that they are clear and transparent, and you can see through them all day, every day. You've heard of a cataracts, yes? That's clouding of the lens, right? And due to either damage because of UV radiation or diet or smoking, those crystal proteins become deformed, and then the lens becomes less transparent, more opaque, so you have to have surgery to repair that. All right. Anyone know what the white of the eye is called up here? Sclera. Very good. The sclera is the white of the eye. It looks kind of blue in my picture, but it encapsulates the entire body of the eye. And the sclera is the site for muscle attachment. So your superior, inferior rectus, your medial lateral rectus, and your superior inferior oblique muscles all attach to that sclera. Sclera is very, very tough, and it's continuous with the dura mater of the brain. So this dura mater, remember that how hard that dura mater was when you cut into that pig's skull last semester? That's how hard and tough the sclera is, and it's meant to be for protection. Now, in the anterior chamber of the eye up here, the sclera is continuous with this area called the cornea. Now, the sclera and the cornea are connected. It's the same tissue. The big difference, though, is the sclera is opaque. The cornea is clear. You're looking through your cornea right now. And externally, spelled that weird, externally, the cornea is stratified squamous epithelium. Internally, it's simple squamous. And this simple squamous on the inside of this cornea has specialized sodium pumps. And the action of those sodium pumps are continually pumping out ions out of those squamous epithelium. And the action of those pumps help make the cornea clear. Without those pumps and without the careful arrangement of the cells in the cornea, we'd be looking through a haze all day, every day. So thankfully, we've got those specialized pumps that help us have good clarity in our vision. Now, the other thing that's unique about the cornea has no blood vessels. Zero blood vessels. And you can see this because when you look at me, am I red? Well, I might be pink, but like, am I like blood red? Hopefully not, right? That we just be all living in a Game of Thrones episode all day, every day, right? When you look at the world, you don't see this spider web of blood vessels obstructing your vision because there's no blood in the cornea. That means the cornea relies on all of its surrounding tissues to get its nutrition. There's no way for it to get. The other thing that makes that cornea unique, because it has no blood vessels, this is the easiest tissue to transplant from person to person in the body. This is the easiest tissue to transplant. I could give any one of you my corneas right now, and your body would accept them, no problem, because there's no blood. There's an identifying feature that makes it mine. Does that make sense? So corneal transplants are very simple and very easy and restore vision to those who have been blinded by injury or other things. So it's a wonderful thing that we figured that one out. The last thing that's true about the cornea is it's highly innervated. Lots of nerves. And mainly these nerves are pain receptors. Think about the last time you shoved a mascara brush in your eyeball, right? And you hit the floor, and talk about lacrimal secretions. Yes, it's painful. The cornea is heavily innervated. The sclera is much less so. You can, if you're of strong stuff, you can look in the mirror and touch the white of your eye. But if you slide that finger on the center of your eye, no dice, right? You're out. Absolutely not. That's because of all of the nerves in the cornea. Why do you think we have so many nerves there? Absolutely. This is the really vulnerable part. It's the most anterior portion of the eye. This is what we see through all day, every day. Do not want to injure it, so we have tons of nerves present. So these two structures together, the sclera and the cornea, make up the fibrous layer of the eye, which is the toughest, biggest protective layer of the eye. So let's move kind of anterior posterior. So this chamber in the cornea, this is called this is the anterior chamber. It's filled with aqueous humor. Which is just a saline-like fluid that's going to circulate here in front of the lens and behind the lens and in front of the lens and behind the lens all day, every day. Do you know when you go to the eye doctor and they puff that air in your eye and you jerk dramatically and blink for a while? They're testing the pressure of this fluid in this anterior portion of the eye. If the fluid inside the aqueous humor fluid doesn't circulate well, it can cause the cornea to bulge and pressure to increase in the eye. Have you heard of glaucoma before? Yes? Can cause blindness if the pressure increases to a point that we're obstructing all the blood vessels back here in the back of the eye. All right. Now, this space right here, anybody know what that is? pupil. The pupil is actually a non-structure. It's a hole. There's no structure that makes the pupil. It's just this space. And when you look into the pupil of someone's eye, what you're really seeing, that dark color that you're seeing, is the back of their eye. You're looking all the way through the cornea, pupil, lens, and you're hitting back here on this retina there. And that pupil is what is allowing light into the eye. Pupil size is controlled by the iris. And the iris is the pigmented portion of the front of the eye. You might have a brown iris, you might have a blue iris, you might have a green iris. But the iris is basically 2 layers of smooth muscle. Smooth muscle, is that voluntary or involuntary? Involuntary. So can we control our pupil size voluntarily? No, it happens. I can't anyways. You might be better at it than me. Yes, ma'am.
So when they dilate your eyes,
Yes. Yes. So there's, can I answer your question in a long way? Okay, there's 2 muscles. There's the sphincter papillae, or pupillae, and the dilator muscle, dilator pupillae. When the sphincter is contracted, it decreases pupil size. When the dilator is contracted, what do you think it does? Increases, very good, pupil size. So when we dilate the eye, if you go to the eye doctor and they do that so they can see more of the retina, they activate this dilator muscle, makes that muscle widen, and it widens the pupil size. Both of these are under autonomic control. The sphincter is parasympathetic. Thought we were done. And the dilator is sympathetic, but it rears its ugly head every time you think you're done. Yeah. Now we've got pigment in our eye, in the iris. What's the main pigment of the human body? Anyone remember? Melanin. And again, this is melanin pigment in the iris. Whether you have a blue eye or a green eye or a hazel eye or one of each or brown, all the pigment that gives your eye its color is melanin. It's the only pigment. Several genes code for eye color, but what makes your eye the color it is what light is reflected back. So the less melanin you have in your eye, the lighter color your eye is. And those of you who have low levels of melanin in the eye, so blue eyes or green eyes, are you super light sensitive? When you go outside and the sun is out, you're like, blah, I gotta have my sunglasses on immediately. That's because you don't have that much melanin. Melanin absorbs light and prevents light from bouncing around in the eye. So if you have a light eye, that light hits your eye and starts bouncing around and is really blinding to you. It can cause you to squint, and so you grab your sunglasses because the sun never sets, you know what I mean? And that is helpful to you. Okay, wonderful. Moving backwards. So this lens, do you see these little teeny tiny attachment points around the, how about now? Yes? Yes? Okay, and then here and here, there's this little tiny meaty muscle. And that muscle is called the ciliary body. Some texts will call it the ciliary muscle. It's a smooth muscle. And its job is to control the shape of the lens. Oh, you can't see that. There we go. Okay, its job is to control the shape of the lens. So if you look, this ciliary muscle here has all of these little tiny ligament attachments are called suspensory ligaments. Did you ever play the game in grade school where you all grabbed a hold of the parachute and you rose it up and you popped the balls or whatever, and someone kicked the chosen one got to sit in the center and get lifted up in the air? That was never me, but I always wanted to be that person. Suspensory ligaments are like you holding on to the arms holding onto the parachute. So circling this lens are all these little tiny ligaments holding on to the lens and attaching the lens to the ciliary body. Now, whether or not the ciliary body that muscle is contracted or relaxed, it changes the tension pulled on the lens. Yes, ma'am?
So you having astigmatism in your eye, is that the ciliary body?
That's a great question. And astigmatism actually has very little to do, astigmatism is a misshapen lens. So someone who has astigmatism, like I do, their lens is not the normal kind of football shape. It's either too flat or too fat, depending on what color. So your suspensory ligaments could pull on that lens all at once and not be able to focus light onto the correct part of the back of the eye. So we do corrective lenses and help adjust light as it flows in and make sure the light hits in the right spot. But someone with an astigmatism, your ciliary body muscles physically can't contract enough to change the lens shape enough to make it focus in the right spot? That's a great question. So speaking of that lens, so if you're looking at something close, the lens is going to bulge. Did I spell bulges right? That word looks really weird. Okay. Close vision, the lens is going to bulge. We can and loosen the ligaments and the lens bulges. For far vision, I'm sorry, you can't see that word. Far vision, lens flattens. We relax the ciliary muscle. And the lens flattens. So all day, every day, the ciliary body is contracting. Those suspensory ligaments are putting tension on the lens and changing its shape so that we can focus light on the perfect back corner of the eye, the area of sharpest vision. Alrighty. Okay, so moving anterior, posterior, we're sliding to the back of the eye now. So if you look underneath this sclera runs all the way around the back of the eye, but there's this layer kind of almost looks like it's got speckles in between the choroid and the retina. In between the sclera and the retina is called the choroid. This is a heavily pigmented layer. So what do you think it has lots of? Full of melanin. Tons of melanin. What kind of melanin? Blue, green, brown. And the job of the choroid is to absorb extraneous light. Tons of light's coming in our eye all day, every day. And any extra light, if it wasn't absorbed, would bounce around in there, engage photoreceptors in odd places, and we would only see shapes or movement or size or color. It would be a hot mess. So the choroid absorbs all that extra light and helps us focus. And the big kind of where the magic happens in the eye is this innermost layer. It's called the retina. The retina is kind of the big deal in the back of the eye. It's the location of millions of photoreceptors. There's lots of glial cells, neural glial cells, and processing neurons. There's two real layers of the retina. There's an outer pigmented layer, which is fused with the choroid. So it's got lots of what pigment in it? Melanin. Yes, ma'am, Christina? Do you have a question?
It was after the choroid.
Mm-hmm.
So it's like, you know, like a Super Bowl, and you close your eyes, the color that you see from the choroid.
So can I save that one? What happens, your photoreceptors are bleached in that it takes some if you've got a bright light and you blink, and all you can see is that light. Or if you're driving at night and the headlights go by and you keep blinking, you still see those doggone headlights, right? What has happened is all of your photoreceptors have been engaged and it takes them a minute to recover, and you're noticing that pause of recovery time. I'll come back to that one. That's a good story. Okay, so this outer pigmented layer is full of melanin. It's also got lots of phagocytotic cells, phagocytes. They're going to be debris cleaner-uppers, so you've got lots of debris that's formed when we do vision, and vitamin A storage. Your mama ever told you to carrot so you'll see good? Or your grandmama? She's not wrong, because vitamin A is really helpful for vision. But the big layer that we really focus on is the inner neural layer. And that's where all the photoreceptors are. Now, the main photoreceptors in the eye, anyone know what they are? There's two cell types, rods and cones. Very good. Rods and cones. Millions of them found in the back of the eye, in the retina. Now, if you'll notice, the back of the eye has lots of blood vessels in it. Those blood vessels are actually in that choroid and in the retina, providing all kinds of nourishment to these photoreceptors inside the eye. Now, if we looked at this retina, this kind of pink, this whole area here is the retina. There is a tiny spot right here that you can kind of see in your picture. Sharpest vision. It's called the fovea centralis. The fovea centralis is the point of sharpest vision. Everything the eye does is meant to focus eye on the fovea centralis, to drive light right here to the back of this eye, to put that light right there. So everything from the cornea to the aqueous humor to the pupil to the lens to even the vitreous body, which is the gel substance that of its bulk. All of those structures, vitreous body, are focusing light right back here on the fovea centralis. It's the point of sharpest vision. It's the point where we have the most photoreceptors in the eye. And specifically, we have only cones in this part of the eye. Cones are our photoreceptor that can see sharp vision, bright light vision. So we have tons of cones in that area. If you kind of draw a dotted circle around that fovea, this is an area called the macula lutea. And as we radiate out from the fovea centralis, we decrease the amount of cones that are present. So if the highest point of cones are here, we radiate outwards. The macula lutea is mostly cones, not all cones. And you slowly get a few rods sprinkled in. By the time you get to the outer edge of the retina, here or here, what do you think we have? Not mostly cones, but mostly rods. So the further you get from the fovea centralis, the more and more rods that you get here. All right. Now, all of these photoreceptors in the retina are eventually going to converge onto this cranial nerve right here. Cranial nerve number, it's the optic nerve, and it's anchoring the eye in the back of the skull. And this optic nerve is eventually going to go to the optic chiasm, which you saw in lab last week, cross right to left, and send signals to the brain, to the occipital lobe of the brain, which is our main visual processing area. And do you notice there's an artery that is associated with this optic nerve? That's going to be your central artery and vein. Its main job is to supply good oxygenated blood to the eye, drain wastes away from the eye all day, every day. Now, because this optic nerve is the place where all of the photoreceptors are converging, you can think about all of those nerves running and nose diving down into this optic nerve here. This area right here is the only place in the eye without photoreceptors. Instead, you just kind of got the butts of the photoreceptors that have already dived down and gone towards the brain. We call this the optic disc. It's your blind spot. You legit have one. You never notice you have one because each eye compensates for the opposite eye blind spot. And we'll find this blind spot in lab. But it is the only point in the retina that has no photoreceptors. So are there any rods here? No. Are there any cones here? No. Just the back ends of, I say dive down. What I want to do is zoom in on the retina, and let's look at the specific cell types that are found there. So if you could, this picture is the next one. You could pull out this image. I want to show you the cells that live in the retina and what they do. So this is just a tiny cross-section chunk of the retina. And we're spending a lot of time on this anatomy, but this is just fascinating stuff that happens in the eye. We're turning light into action potentials here, and that is just a wild and crazy thing. So we said the retina had two layers, a neural layer and a what else? Anyone can remember? Those are the P. Pigmented. This teeny tiny layer right here is the pigmented layer, and this big thick layer right here is the neural layer. We said the pigmented layer was full of what pigment? Picture all these melanin granules here absorbing extra light that we don't need, right? Now, the neural layer is where you're going to find all of your photoreceptors, all of your glial cells, and then ganglion cells. So these blue and yellow cells down here, those are your photoreceptors, polar cells. So they're nerves. And these white cells here are ganglion cells. And so vision is this great game of telephone. The photoreceptor sends a signal to the bipolar cell, which sends a signal to the glial cell, which then eventually is going to converge and send a signal via cranial nerve number number 2. Right? And then along the way, there are cells that help modify the signal that's being sent. And both of these green cells are those cells. This dark one is an amacrine cell. And this light green one is a horizontal cell. Aren't you? Such an interesting name. And both of their jobs is to help focus the signal. Help focus the signal. Both amacrine and horizontal cells help focus the signal. Yes, ma'am?
On the PowerPoint.
Either one doesn't matter. Yeah, either one, it doesn't matter. They're both amacrine and horizontal cells. They just colored them that color. Yeah. Either way, it does not matter. So these rods and cones, let's look at them. Anyone want to guess which one is the rod, the blue one or the yellow one? Yeah, the blue one is the rod, and the yellow one is the cone. These are our two main photoreceptors. Rods work best in dim light. They're mainly used for peripheral vision. Do you watch The Office? Anybody? When Michael Scott's like staring forward and using his peripherals? Anyways, entertaining. Okay. And they increase in number. They're high in number towards the edge of the retina. All right. So these we usually say are for our black and white vision. The best way to describe how the rods look when you activate the rods is whenever you cut the lights off in a room and you're looking around, everything is in kind of this gray tone. You can still see fuzzy. That's the best way to describe our rods and how our rods work. Our cones are our color vision. These are our high resolution photoreceptors. And we actually have 3 different types of cones. We have red cones, we have blue cones, and we have green cones. So really, in total, you have 4 different types of photoreceptor cells. You have rods, red cones, blue cones, and green cones. Now, each of these photoreceptors are responding to a photon that's moving in a wave through the air. We don't want to get too deep into physics here, right? But each photon moves at a different wavelength. And so by saying it's a red cone or a blue cone or a green cone really means that each of those cones responds to a different wavelength of photon that's moving through the air. So a red cone is going to respond to a photon moving at a 560 nanometer wavelength. A blue is 420. Rods respond to about 500 nanometer wavelength light. And the reason why we can see such a beautiful variety and array of colors is because there are overlaps in between which of these cones are sensitive to what, right? And so different photons moving at different speeds or wavelengths are going to activate several cones and several rods, and that's what gives us teal and magenta and fuchsia and sepia. Yes, ma'am. I'm making it. That's a color, right? Yeah.
Why the cones not the primary colors? Why is it?
Why not yellow? We see yellow because of the overlap, and mainly it's the lower end of the red edge. But I don't know why we don't have a 4th one that is for the other primary color. We decided what the primary colors were, right? And that you probably need to talk to like Van Gogh about that. I'm not sure who was involved in that conversation. But we don't have a cone that's specifically for yellow. And in the animal kingdom, tiny tangent, different animals have different types of cones. For example, fun fact, whales have lost 2 of their cones and only have 1 left. Which one do you think is what whales have? Blue. Because all they see every day is light that's refracted by water, right? And that water bends the light in such a way that it only gives blue wavelengths. They just see blue, which would be kind of cool and wild. But yeah, anyways, I'm not sure why we don't have a yellow one. But the fact that we have 3 is kind of cool. 3 is the most that you can have in the vertebrate world. I mean, if you're an insect, you can see in the infrared, which is why it doesn't matter if you're wearing blue jeans or not, that mosquito is still going to bite you. And if you're a bird, you can see in the ultraviolet, which is also just very cool. But fun facts. Okay, moving on. So each of these rods and cones, do you see how they're kind of nose diving into this pigmented layer? I'm sorry, yes, ma'am.
So for those colorblind,
Ooh, yes. If you are, the red-green colorblind is the most common, right? It's an X-linked inherited congenital disorder, but you've lost one of your cones. You can still see color, you just can't discriminate that particular wavelength of light. And so you have a hard time discriminating between really close wavelengths of light. But yeah, great question. So do you see how each of these rods and cones is kind of nose diving into this pigmented layer? And they have this kind of extended beak-looking structure here, the cone and then the rod. Each of those noses of those cells are layers and layers of discs filled with visual pigment. And the visual pigment that we use to see is called rhodopsin. And rhodopsin is a big protein. It's made up of 2 things put together. It's made up of opsin plus a retinol, a molecule of retinol, which is derived from vitamin A. And if I were to draw it, I'm going to give it a try. It kind of looks like a dumbbell. Sad dumbbell, but you get the idea, right? The dumbbell part is the opsin, and the retinol will be inside here. It kind of looks, it's got this hexo sugar and this fun little tail. That's the retinol. It's this big protein molecule. All of these discs will be filled with this rhodopsin. And every single time light hits the rods and cones, this rhodopsin is excited, and that causes a signaling cascade to send a signal from the rod or the cone to the bipolar cell, to the ganglion cell, and then down through cranial nerve number 2. And we'll look at that in just a second. But all day, every day, light is degrading these discs. And so you're constantly synthesizing new discs. Every 24 hours, you get a whole new stack of discs filled with rhodopsin. So if you don't have a lot of vitamin A in your diet, you don't have the ability to rebuild those discs. So vitamin A it really is really important for vision and seeing. Now, before we get into the molecular side of what happens when light hits this rhodopsin, I do want us to notice a little thing here going on in the retina. So look at your rods and look at your cones. Which has a 1-to-1 ratio of photoreceptor to bipolar cell? Cones or rods? 1-to-1? Cones. You just didn't say the other one, right? Yeah, so there's 1 cone to 1 bipolar cell. Do you see that? How many rods do we have to 1 bipolar cell? 3, or 2, or 3. We called that, way back when, convergence. Right? When many neurons synapse onto one, we call that convergence. So what do we see convergence in? Rods or cones? Rods. 3 go to 1. That's convergence, right? 1-to-1, that's not convergence. The reason why our cones are our sharp vision is because there's a 1-to-1 ratio of cone to bipolar cells. Rods, we have fuzzy vision because that signal gets filtered down and combined with all of the cells around them. Are you with me there? So that convergence is what gives our cones the ability to have that solution. So rods converge. Many rods converge onto 1 ganglion cell. The next piece of the story, we have to look at how this whole process comes to be. So this is the next picture here. So let's zoom into what's happening to this retinol when light hits it. All right. Vision is just too cool to not look at this stuff. My drawing wasn't terrible. See the barbell shape here and the little guy in the center? Yeah? So this big barbell, what did we say that was? We called it the opsin. And inside is the retinol. Very good. Very, very good. All right. So follow along with me in this picture because this is going to get back to Christina's question about why do I see things after I've seen them, right? So first step 1, light hits rhodopsin. And this rhodopsin is either going to associate with a rod rhodopsin or it's going to be blue, red, or green opsin molecules. And that light hits that rhodopsin. And it changes the retinol configuration from a cis, biochemistry, to a trans. This little hitch in our giddy up to a straight configuration. So 2, change from cis to trans. Okay. We'll pause here for today. I hate to stop. There's so much more fun things to say. Please make sure you practice the structures of the eye, what each one of them does and means, and the different layers of the retina. We'll come back to this on Friday. Have a great afternoon.